Hello and welcome from Milan, Italy. We've had a number of wonderful updates and meetings to do with our International Progressive MS Alliance. It's wonderful to be here. My name is Matthew Miles. I'm the CEO of MS Research Australia. I'm also the Vice Chair of the Executive Committee of the International Progressive MS Alliance. In a moment, I will introduce our esteemed panel, but I just wanted to start by going through a few important points about the International Progressive MS Alliance. This is really a global collaboration of MS organisations and researchers, our industry partners, donors and supporters in an unprecedented global collective. There's more than 18 countries that are involved in the Alliance, and together we're working to make sure we can provide solutions for people living with progressive MS. We wouldn't be able to do the work we do without the wonderful support of our partners for this webcast, this global webcast. So I'd like to thank our supporters, Novartis, and I'd also like to thank Genentech, another one of our supporters. It's greatly appreciated, their work. As we go into the webcast, I'd like to also recognise why we're here. Over 2.3 million people worldwide live with MS, and it's estimated that over 1 million people are living with progressive MS. This is a difficult, challenging disease to combat, and the International Progressive MS Alliance is about bringing the best people in the world, the best minds, to try and find those solutions, to cut through the barriers, to ask ourselves the really difficult questions about progress in progressive MS. And it's been wonderful over the last couple of years to see some of that progress come to fruition. But effectively, more must be done for people living with progressive MS. We're very, very clear on that. We know that there's been almost unprecedented improvement in the lives of people with relapsing MS, but we need to make sure that that progress is something that benefits everyone and not just some. So today we're going to talk about some of the work of the Alliance. We're going to concentrate on one of the network awards and one of the really important collaboratives that we fund as the Alliance. And we're going to talk about some of the ways that we might get treatments to people living with progressive MS. In terms of investment, this sort of progress doesn't happen just because it's fair or it's right or it's just. It needs money, it needs funding. And the funding side of the Alliance is absolutely critical. And together, as a collective, we've invested more than 60 million euros into this type of progressive MS research to fund the best in the world so we can find those important solutions. So without further ado, I'd like to introduce our wonderful panel. And I'd like to start by introducing Professor Gianvito Martino. And Gianvito is one of the world's leading MS academics, and he works at a local institution in San Rafael here in Milan, and is leading one of the network awards for the International Progressive MS Alliance called the Brave in MS Network. Gianvito has published more than 250 important publications and has been absolutely critical in the management and treatment of MS. So welcome, Gianvito. It's wonderful to have you here.
Pleasure to be here, Matthew. Thank you very much for the kind words.
I'd also like to welcome Professor Tanya Coleman, who is from the University of Münster in Germany. You also are an important collaborator in the Brave and MS Collaborative Network. You're a neuropathologist, and we're going to talk a little bit about that a little bit later on in the webcast. But your work is really key in understanding the effect of MS on the myelin in demyelination, but also remyelination. And that's something that I know a lot of people with progressive MS really want to know a lot about. So welcome and thank you for joining us.
Yeah, thank you very much. It's a pleasure to be here.
Lastly, I'd also like to welcome Dr. Tim Coetzee. Tim is got a very wide portfolio at the National MS Society, is heading up not only the services advocacy, but also the research side of things at the National MS Society, but importantly is playing a really key role in the International Progressive MS Alliance. Tim is a great strategist, a great thinker, and with a lot of colleagues in the Scientific Steering Committee is really trying to think and plan out what we do as the Alliance, what are our priorities, and how do we get past some of those hurdles that have challenged us over the last decade or two. So welcome, Tim.
Thank you, Matthew. It's great to be with you on the panel.
If I could just start by asking to you, Gianvito, with the Brave and MS Collaborative, what's the big idea that you're trying to solve with this important global collective?
But the network we are working with is dedicated to find new therapies for the progressive MS. And the idea we had at the beginning was that we have a lot of different molecules and drugs which have been already developed and theoretically could be used already in humans, which maybe have been developed for other diseases or have been abandoned for many reasons, economical ones or whatever, and those are called, and now we know that we can repurpose these drugs and try to understand if those drugs can be also effective in other diseases than the ones for which those drugs have been developed. So we have around 5,000, 6,000 molecules of this type. So the idea was to try first to understand using an in silico system to filter out which of those molecules could fit with the different characteristic of MS. So the idea was to filter the molecules which theoretically could be useful in this disease, and then develop and set up a pipeline, a platform, going from in vitro study to in vivo study in order to understand which of those molecules could really be effective in promoting remyelination on one side and in protecting neurons and doing the so-called neuroprotective effect. This is the simplest thought we had at the beginning, which is not particularly innovative, but the point is that, and the beauties of the system is that we collected the best scientists working on this in this field in the world, and we collected the different arrays which we put all together in order to avoid false negative or false positive. So the platform is really making the difference. Within those platforms, of course, we are using also the so-called induced pluripotent stem cells, which are stem cells that we can get directly from MS patients, in particular from skin biopsies, and in vitro we can transform them into oligodendrocyte or neuron, so to be much closer, possibly, to the disease in itself.
And obviously the idea is to have a number of molecules that you'll be testing. So for the people at home, not every molecule that we test will eventually become a drug therapy. So it's important that you've got a number of molecules, isn't it? So not just one or two, but many.
That's a point, Matthew, actually, because, developing a drug, it's a time-consuming and very costly process. Until a few years ago, developing it was, people was used to say that among 10,000 potential drugs, after 20 years and spending $1 billion, you could get to have one on the market.
Yes.
So very, very difficult and tough story. So we decided to take this approach in order to shorten the period in which we can go from in vitro study to human studies. And that's the reason why we decided to select drugs which are already developed and which have been already used in phase 1 trial, the so-called safety studies, and therefore this is a way to speed up everything and to get to 4 or 5 potential molecules at the end of the project to test into patients.
Right. And Tim, when we were looking at these big network awards, it was incredibly competitive, wasn't it? There was, I think at one stage, probably 500 MS researchers who were involved in putting in applications for these big awards. Gianvito's Global Collaborative was one of those big network awards. What was it about it, do you think, that made it so important that we funded this incredible work?
Well, that's right. You touched on some of the key aspects of it, and I think that first to say that the reason that the Alliance exists is we need more treatments for people with progressive MS, and we had heard the global call from people affected by this form of MS to really address the gap in new treatments. But Vito also touched on the enormous challenges that we face in getting new treatments to people with progressive MS. And so what inspired us was we put out a call saying, we want the best ideas. We want collaborators working together because we believed in the power of teams coming together to really synergize and to create a special environment in which to accelerate the process. But in evaluating these also, the things that we looked for were, is there innovation? Is there a strong team? Does the team have the leadership that you need in order to make the choices to drive the program forward? But also another element is vision, and vision to see the possibility, and vision to really see the program through that brings it all back to why we're here, which is people with progressive MS. So when you take all of those together, Gianvito's was one of the programs that really stood out for us as having all of those dimensions of collaborative effort, experts working together with a clear vision and the kind of global leadership you need in order to have the impact we need. And so that's what led us to that selection.
And people living with progressive MS often ask about how much collaboration is there throughout the world? Are we replicating work, or are we truly working globally together? And your network is probably a great example of that global collaboration, people working in many different countries. Can you tell us a bit about the researchers and what they're doing and where they're from?
The idea we had at the beginning, because scientists are strange people because they are used to work, at least until a few years ago, they were used to work alone, and this is, as Tim was alluding before, this is not anymore the way to make science. So we decided that we could not only rely on just one method to try to discover those drugs, and therefore we thought that putting together more than one method developed by great scientists, which had the possibility to use this method, technically speaking, in the best way possible, was the way to avoid to try to get the wrong way and to get at the end of the process with a drug which was not tested exactly as it should be. So we decided to select method, the best one possible around the world, and then to put those people developing those methods and being really expert in this together in order to avoid, as I was saying before, to avoid that we could get the wrong way and we could not test or discover the right drug that we were aiming at. This was so, as Tim was alluding, what was the major reason why we decided to work together is that we were synergistically and complementary each other. We were not overlapping one to the other. So we are all doing different things, but putting all those things together, we are much, much stronger than working alone.
And I'm assuming you've got, with that global collaboration, you've got a lot of early mornings and late nights to make it all work together so you can all talk together and update each other on all the progress that's going on.
It's a big network. There are people from US, there are people from Canada, from Germany, from France, from Italy. So we are really a global network, and I really appreciated my colleague to accept the invitation to be part of it.
One of the things that we've done is that some of the Alliance staff have had the chance to go through the laboratory and look at the
Sure.
The research in action, and I know that we've got a short video on some of that work. So that's very, very exciting, and thanks to Paola Panini, who was really hosting that work. So I wonder if we could just show that short video on some of the research that's happening here in Italy.
We are now in the laboratory of neuroimmunology. We are at San Raffaele Scientific Institute. This laboratory is focusing its research on the study of multiple sclerosis, and in particular the focus of the study, the main project we have at the moment is a project which is fully supported by the Progressive MS Alliance, and the name of the project is Brave in MS. And we are indeed brave because our focus is to develop new therapeutics for our still unmet clinical need, which is the progressive multiple sclerosis. To this purpose, we are trying to generate oligodendrocytes and neurons from induced pluripotent stem cells with the aim to test new drugs or new therapeutic entities on these types of cells which are critically affected in the progressive multiple sclerosis.
That was fantastic. Thank you. That was a wonderful, wonderful video. And I wonder if I could go on to Tanya and to hear a bit more about your work. You're a neuropathologist, and a lot of people watching the webcast may not understand what that is. And I wonder if you could just talk to us a little bit more. What does a neuropathologist do? And tell us a little bit more about the research that you're doing.
So I'm a neuropathologist. That means I'm a pathologist specialized in the disease of the brain and the spinal cord. So for example, if someone has a brain tumor, the neurosurgeon will take out the brain tumor, and we as neuropathologists, we get the tumor and we analyze the tumor under the microscope. And then we can say what kind of tumor that is. And it's important because this is important for the subsequent treatment of the patients. However, it's not always a tumor. Sometimes it can be also infectious disease or, for example, MS. So we have a chance to analyze MS lesions under the microscope, and that is very helpful because then we understand what is going on in such a lesion. And for example, my group and others, we could show that there's limited or insufficient remyelination in MS lesions, at least in a subset of lesions. And the question that we want to understand is, why are some lesions that remyelinate very well, but others not? And this is actually the basic question of my research. And we try to address these questions, for example, with these iPS-derived oligodendrocytes Gianvito was talking about.
And so with a neuropathologist, you'll look at both tissue under the microscope, but also things that you can see with the naked eye. And I'm assuming it's important to be able to see like what's normal, and then particularly with MS, what's abnormal, so we can really understand some of the basic mechanisms of MS. Yeah, thank you very much. Thank you. Gianvito, a lot of people ask around this idea of repurposed therapies. We hear a lot of that at the moment. What are some of the advantages when we're thinking about repurposed medications and their use in potential use in progressive MS?
The major advantage is that those molecules are ready to go into humans. So of course this makes the process much faster in terms of delivery, new treatment. And the point is that, there are really a huge amount of molecules that can be repurposed to some extent. And that was the initial idea we had. However, you don't know which of those molecules could fit really with MS, with progressive MS. That's the reason why we decided to, Sergio Baranzini in UCSF, he developed this spoke system, which, as I told you before, it's an in silico system in which he put all the information possible about MS, about autoimmune diseases, about myelin, about axons, about neurons. And then, using those kind of information, he filtered out those drugs among the repurposing ones which could fit with the data within this in silico whatever database system. It's very complicated. I'm not a bioinformatician, so I cannot tell you exactly the details of the machine and how the way the machine works, but as a matter of fact, we end up with 1,500 molecules. And then through this system, I make it shorter because it took 2 years of work, we have now a list of around 30 molecules, which after being screened through the in silico system have been tested in vitro on oligos, both human and mouse oligos, and neurons, both human and mouse neurons. And in the system, as I told you before, we have been developed, and we could really measure remyelination, demyelination, neuroprotection, or neurotoxicity. Those 30 molecules have been shown to be the best possible ones. So we didn't think really to be so lucky. I shouldn't say lucky, but also, I mean, probably the work that we designed was done in the right way. But we are very happy because at the end we shortened the list, but this list is not is ready to be tested now in vivo. Of course, in animal models of the disease, more than one, which is important because, as you know, there are no animal model resembling MS. There are different models, and each of them is representing a part of the story, not the entire story, so we have to use more than one. But the design was solid enough to get to enough molecules to be ready to be tested in animal models. Mm-hmm. So I think that the prediction we had at the beginning is probably right. We don't know, of course, out of those 30 molecules, which of those will be really ready to get into humans soon, but certainly the next step would be to understand which of those could be really tested in humans. And as soon as we have demonstrated that some of those molecules do work on animal models, we are ready to go into humans. So the process is much shorter than what we thought before.
And when we talk about repurposed molecules and repurposed potential therapies, is it fair to say that those potential therapies may be a little bit quicker to get to market, where we can be sure that they can work in people with progressive MS and potentially be a bit cheaper? Is it fair to say?
This is something difficult to say because, you know, it's very much depending on different things. I mean, because most of the molecules are based on chemistry. Those are chemical compounds, okay, are not biological compounds. So, of course, chemical compounds usually cost much less than biological drugs. But there is the point is that it depends for which purpose those molecules have been developed. Depends on the reason why those molecules have been abandoned, depends on the intellectual property. Mm-hmm. And so, in my view, of course, this could be a faster and a cheaper way to get drugs, which could be really offered to patients without any major economical burden, let's put it this way. So I think they could be cheaper.
Yeah.
In theory, Matthew.
In theory. So Tanya, how does your work fit into that approach? Tell us a little bit more about that.
Yeah, so as I mentioned, we want to understand why remyelination sometimes works and sometimes fails. So we want to understand the molecular mechanisms. And if we understand these molecular mechanisms, then of course we maybe have a new treatment approach, a new target. And what we are doing, we work with these oligodendrocytes, human oligodendrocytes, and as Gianvito already mentioned, they are derived from iPS. So Gianvito generated the iPS cells, we generated developed protocols to generate oligodendrocytes from these iPS cells. And now we can test these 32 compounds on the human oligodendrocytes, and we can test them on oligodendrocytes from healthy individuals, but also on oligodendrocytes from people with MS. So we can really say, okay, this is a compound that has a positive effect on oligodendrocytes from patients or people with multiple sclerosis. And this is what we are doing currently. And at least I can say we have at least 5 to 6 compounds that also show a positive effect on human oligodendrocytes with respect to differentiation and myelination.
Yeah, and I'm going to ask about that. And just for a reminder for everyone watching the webcast, the oligodendrocytes are the myelin-producing cells. Yes. I know that some people get a little confused on that point. So with the specifics, Gianvito, of what you found with the platform, so can you just talk us through a little bit more about specifics of some of the potential molecules that might be developed into treatments?
What we did, and this should be clear, is that we did everything blind. So all the labs around the world received the drugs without knowing which were the drugs. And this is very, very important in order to try to have solid data at the end of the day. So was the first thing we did. So we started testing those drugs without knowing which drugs we were using. Then, since, as I mentioned before, we are using different assays in different parts of the world within our network, only at the end of the first round, which was based only on testing those drugs on cells from rodents, for the simple reason that when you have a lot of drugs, you need a lot of cells to test them in vitro. And unfortunately, only mouse cells of the brain could be generated in the lab, and you can have only high amount of cells only from rodents. So we started selecting the drugs which were selected by the in silico system I was mentioning on mouse cells in blind. Then we combined all the data and we tried to understand which were the molecules which were positive on all the different tests we have been using. This is important. So only if the molecules were positive in all the tests and assays we have been using, then we gave a go signal and we moved into human cells. Unfortunately, you cannot have too many human cells in vitro to use. So you have to have few drugs to test on human cells. Human cells are very difficult to grow, very precious. We have been lucky enough that Tanya developed a very nice system to produce a lot of myelin-producing cells from humans in few days, starting from skin biopsy, those kind of strange stem cells which can be obtained from MS patients as well. And then we moved into human cells again in a sort of blind fashion, and again, and this was really exciting and challenging, results were overlapping in many tests. And this was very much reassuring, because it was not biased by the test itself, but was really depending on the molecules, on the drugs we were using. So that's the reason why we are quite optimistic.
So it sounds like you've been very encouraged and happy with the progress of the work so far.
We are, I should say, we are happy about what we did so far.
That's great. And I'm sure that people living with progressive MS will be really happy to hear that. There's a lot of work that goes on with the Alliance, Tim, and there's a number of things that we're funding, other big network awards, other teams. So could you just tell us a little bit more about some of the other work that the Alliance is funding at the moment, and maybe just a few updates on progress so far?
Certainly, and I think you've seen in the team that Gianvito is leading the kind of synergy and innovation that we're going for and want to achieve with our other networks. So as you mentioned, we have 3 networks that we're funding. 2 are focused on drug discovery. One is focused on accelerating, developing tools that will help us do clinical trials faster. So one of the networks is led by Dr. Francisco Quintana and another international team that is focused on identifying and understanding the parts of the immune system that are involved in the brain and identifying treatments that can intervene in those immune attacks on the brain and really create a different paradigm for how we can manage the immune system in progressive MS and in effect stop, hopefully, some of the attacks and damage that we see in progressive MS. That team, like Gianvito's, has been very active in discovering new treatments. They've identified a number of candidate molecules, as Gianvito's alluded to, and are working their way through them to try to get to some smaller number of candidates that have the highest potential that we can then move into testing first in animal models and then into people. Gianvito's team obviously leads another dimension of our networks in terms of identifying those molecules that can restore function, either through myelin repair or neuroprotection, so protecting the brain from damage. So these 2 networks together in just 2 years have identified a number of molecules that we can have in a portfolio to create a pipeline of new treatments. But the important challenge is not just finding treatments. We also need to be able to measure what's happening in the brain of people with progressive MS to determine whether or not these treatments are working effectively. And that's been one of our major challenges, is that we've lacked the tools to be able to look and see with precision what's happening in progressive MS. So the other network we have funded is led by Dr. Doug Arnold and an international team of people who are experts in using imaging and MRI, and also technology that comes from the artificial intelligence world of using computing power to be able to analyze images and to really understand what is happening in people with MS by looking through MRIs. This team has assembled tens of thousands of images from clinical trials where people with MS have participated. They've had an MRI taken, and that information is now being gathered into a very large database that the team will then use artificial intelligence platforms in order to try to analyze those and determine whether or not they can see a specific signature or picture that tells us that a person with progressive MS will be on one trajectory or another. And the idea behind that is that that tool, when perfected and validated, could then be used in clinical trials of the kind that Gianvito and Dr. Quintana's team are imagining. And then beyond that for the community, because our goal here is to really create tools that the entire community can use, and through that shorten the number of how long it takes to do clinical trials.
And there's, as you alluded to, there's great synergies between the big network awards, isn't there? They're working really well collectively together. It's not in isolation, they're working together.
Absolutely. In fact, just last year, in May of last year, we brought all three teams together, about 100 people, for a midpoint check of how we were doing, and the teams presented to each other. And that's another way we create synergies, in that, oftentimes people being together in a room, hearing their results from each other, creates and sparks additional thoughts and insights that don't necessarily come when people are working in their individual laboratories. And it's the kind of convening activity and the power of a collective like the Alliance to bring groups together and to really create and see what we can enable to accelerate progress.
Well, I've just got one question for each of you to go, and I just wonder if I could start with you, Tanya, and maybe if you could just tell me what you feel is different about the Alliance and the work that we're doing.
I think there are two things I would like to mention. The first one is, I'm not aware there's another funding agency that would be able to support such a global group of scientists. So I'm not aware of anything else, any other funding agency. And the second is, of course, the Progressive MS Alliance always motivates us to think how to treat people with progressive MS. And this, of course, is helpful for us because it always reminds us what should we be thinking about.
And Gianvito, what would, you're leading, as we've heard tonight, one of the big networks for the Alliance. What would success look like for you?
I've been participating also in the past in other initiatives of this type, which were funded by the National MS Society. But this time, I think that what Tim said before is exactly the point, that the Alliance is trying really to work globally and is trying to select projects which are complementary one to the other. So it would be really fantastic at the end of this first part of the story to have molecules which could be tested in humans using the algorithms that one of the network is developing. Because once we have the drugs, this is not enough, but we have to measure the efficacy in people with MS. And therefore, the idea that I did appreciate actually was to have exactly three different networks which are working independently to some extent, but could share results at the end. So to make real, even from a scientific point of view, even from the scientific community point of view, a global initiative. So really, we need to work together. It's not anymore the time of the one-man show.
Yeah.
Science is multidisciplinary, interdisciplinary, should be done working together and putting together the best possible scientists and giving them exactly the aim to reach. And this is what PMSA is doing. So I'm very, very happy that you decided to start with this initiative. I think you did a great job. And I think this is the way even to educate new generation of scientists to start thinking out of the box with the other scientists around the world, trying to really get the best way to try to find a cure for people with MS.
And people living with progressive MS have been asking for something like this for many years. They've quite rightly asked, well, when is progress going to happen for their MS? When can they expect some breakthroughs and new treatments for progressive MS? So, Tim, what do you think are some of the take-home messages and the prospects of new treatments for progressive MS?
Well, I think you reminded us at the beginning of the nearly 1 million people around the world that live with progressive MS. And I think what you've seen in this panel is what the Alliance and the global MS community has been able to do together in a way that is unprecedented. The MS societies coming together in a way they had never done before to agree on how to fund research together, to identify priorities, to then identify teams that work together in an unprecedented global way. So it's not just societies, but the scientific community, people with MS. I think we have seen in just 2 years, in a very brief period, in what is a very long process, that we already have candidates that are identified, that we have tools that are now in testing for people. And so I think the viewers of this webcast should be really optimistic that not only is there a vision for innovating and bringing new products, but then that we're actually creating the path for those. But the thing about creating a path forward for any of those is that it takes many different people. It takes people with MS participating in clinical trials. It takes MS Society supporting those efforts. It takes innovators like the teams that we've seen in this webcast today. It takes the companies that will develop those treatments. It takes the health systems that are going to do it. And the power of that is when we all work together, we're able to achieve it in an inspiring way and to really make progress for those 1 million people with the disease and their families and everybody affected by it.
And we've often got all of those people in the one room, don't we? So being able to have all of those stakeholders together has been especially encouraging to hear those different views and to hear, how do we work around some of those, significant barriers which we've had with progressive MS for a long, long time.
Indeed.
I hope that's been really encouraging and exciting for people who are watching this global webcast and for people who are living with progressive MS or affected by progressive MS. I would like to very much thank our wonderful panel who've provided some really great updates and insights into accelerating treatments for progressive MS. So I'd like to start by thanking Gianvito. It was wonderful to hear about the Brave and MS Collaborative Award, and congratulations for everything that you're doing.
Thank you very much. It was a pleasure to be here. Thank you, Matthew, for chairing this session and this panel.
To Tanya, it was fantastic to hear more about the collaborations and how you're working with Gianvito and working on the Brave and MS Collaborative as well. We heard a lot more about the role of a neuropathologist, which I think has confused some people, and to understand exactly what your role is and how you contribute greatly to the work of this important network. So thank you for being part of this panel.
Yeah, thank you very much, and it was a pleasure to be here.
And to Tim, thank you for your role in the Alliance. It's incredibly inspiring to work with you and colleagues in the Scientific Steering Committee. It was great to hear you be able to not only talk about the positivity with this network, but to also let the people at home know about some of the other work that's going on with the Alliance and how it's all fitting and working in together.
Thank you, Matthew. It was a pleasure to be with you.
So thank you to everyone who's been watching and listening. This International Progressive MS Alliance is a really important global collective. And in my mind, it's one of the most important things that we could do as people who are trying to find solutions for people living with progressive MS. I don't think there are many other diseases that could say they've got this degree of global collaboration, this degree of global sharing, this degree of working across borders, and this degree of consensus of what we need to do to find those solutions for people living with progressive MS. This is a really complex problem, and MS is one of the most complex diseases that we know. So it's hard work, but the International Progressive MS Alliance is uniquely placed to do a lot of this work, a lot of the things that we've found difficult over the last two decades. The Alliance is taking up the mantle and really running with trying to solve some of those big problems. So thank you for being part of the global webcast. I hope that you have got a lot out of it. Please don't hesitate to look at the International Progressive MS Alliance website for more information, or speak to your colleagues in your country, in your MS society, about progressive MS. So thank you, and please remember that we are not going to stop until we find solutions for people with progressive MS. Thank you and good night.